Dear candidate, dear colleagues, friends, family, members of the reading committee, welcome. I hereby open this academic session in which Pablo-Alexandre Mallaroni will defend his academic thesis entitled Decomposing the Psychedelic State, Insights from the Brain, Behavior and Context. I kindly ask you now to give us a summary of your great work. Thanks.
Dear Pro-Rector, committee members, friends, families, colleagues, and those watching from abroad, I'm very grateful for this opportunity to talk about what I've been doing in the last 5 years, titled Decomposing the Psychedelic State. To talk about the psychedelic state, we have to define psychedelic drugs. So these are substances like LSD or magic mushrooms or mescaline that, when taken, produce an altered state of consciousness, one that's characterized by feelings of unity, the emergence of a third-person perspective, the blurring of bodily boundaries. And these have kind of captivated science for a while because a 1 or 2 time administration produces feelings of, long-lasting feelings of mental well-being, psychological flexibility, feelings of mental clarity and positive mood. And this idea of flexibility kind of extends in the brain. It's mirrored by it. So if you see the brain as like a set of fixed patterns of connections or functional connections, following the administration of one of these drugs, the brain is more integrated or more flexible. And this is maximal during this acute psychedelic state and may endure due to the presence of neuroplasticity, so the brain's capacity to rewire and reorganize following a stimulus. But, you know, these drugs, it's not all sunshine and rainbows. You know, sometimes people don't really get much out of them, or sometimes people feel much worse and develop mental health conditions. And science over the years has said that the quality of the experience is really matters. So sometimes people, as well as the good, they can also experience feelings of paranoia, fear, delusions, you know, this fear of letting go. And there are other elements, just like sensory components, like the walls are moving, I see things that aren't there, or I hear things that aren't there. So trying to understand why people go in different directions to basically have a lifetime of mental growth or decline is why we need to kind of understand the elements of these experiences. And to do that, we have to kind of decompose the building blocks of these. So what matters could be the context in which you take the drug. It could be, you know, whether you take it alone or meditate, or whether you take it as part of a group, like some religious practices, which is more communal. And this kind of all adds up in certain psychedelics, like ayahuasca, which is basically a botanical brew from the Amazon, which contains the psychedelic DMT. As well, it can be the drug that you take. So whereas spoken about the classics so far, chemistry has shown that different psychedelics can be broken down according to their structure, with tryptamines and lysergamides like LSD that exist within that class, but also phenethylamines, which are derived from mescaline. And chemists over the years have figured out how to play with these chemical structures to produce novel psychedelics, and each of these substances are reported to produce their own types of experiences and varieties of effects. Also, we can't forget the pharmacology of the drug, how the drug works. We know that the brunt of the psychedelic experience is due to this agonism of the 2A receptor, the activation of it, but there are many other receptors at which these drugs work at. And if you see the 2A receptor as like the grand piano in an orchestra, then all of these secondary receptors could be like adding to the symphony of that experience. And lastly, the individual. So you and I, from the genes we carry and everything that makes us us, from the receptors they encode to the neuroarchitecture that it produces, we are all unique. And what neuroimaging has shown is that we can spot one another from just a brain fingerprint, so basically how your brain organizes itself. And understanding each of these elements is important for the individual because some of these things we just can't control. And the questions I'm asking today are not new, but maybe the way we approach them should matter. So typically in science, we tend to aggregate data from large groups of people to try and understand an effect, how something separates. But if I've told you that people themselves are unique and go in different directions, maybe we need to focus on the individual. And for this, neuroscience provides a lot of tools. We can pick between things that have a close relationship with like the mechanisms of these drugs or other things that tell us more about the experiential quality. So it can be looking at the structural properties of the brain, the receptors, where they are distributed in the brain, or the functional connections I was telling you about. For example, looking at their average relationship with one another over time, but also how they dynamically evolve throughout time. But also the regions themselves, so how the signaling behavior of each of these regions can change, how complicated or predictable or entropic they can be. We can also look at cognitive tasks, so the building blocks of behavior, like memory and attention. And lastly, the highest level of relationship to behavior, the questionnaires, so the phenomenology of the drug. And to know why someone goes a certain way, we have to integrate each of these levels of organization. So this kind of led me to ask a few questions. First of all, do different drugs actually do different things? Do different psychedelics pose different benefits and risks? And if different psychedelics work in a different way, because if function comes from structure, then maybe the pharmacology will change the way these drugs affect the brain. And this context, how we take the drug or with what actually matter. And lastly, is there something in each of us that can tell us where someone's going to go? So in chapter 2, we asked 1,000 people about their experiences with novel psychedelics. And here we saw that the quality of the experience was quite important. So first of all, people reported very different side effect profiles, with phenethylamines having a greater propensity to produce physical side effects like seizures, heart problems, and things like this, whereas tryptamines, people reported them to give them more sort of anxiety and paranoia. But at the end of the day, if each drug is unique, then averaging everything within a structural class can actually diminish us finding important differences. So here what we did is we tried to understand whether there was something about the experiences that could distinguish these drugs by picking a representative of each class. And what we saw using a classifier that basically learns from the data, we saw that this quality of losing oneself was really important for distinguishing these three members. So the good and the bad of it, with drugs like 2CB showing less of this ego dissolution or loss of self. And that raised even more questions. What is 2CB? Because it's been around for 50 years, but no one's ever really studied it. And it seems to be a phenethylamine, which I've rarely ever studied. So we took it to the lab to try and understand what it actually does by comparing it to a tryptamine, psilocybin, which is magic mushrooms. And here people were given on separate days a matched dose of 2CB and psilocybin, or placebo, basically. And on each of these days, we tried to get a full snapshot of what this drug does across different levels of organization that I was telling you about. And what we saw was that in a clinical context, not in the wild, 2CB was actually quite interesting. It was safe. People tolerated the drug well. It didn't produce greater incidences of heart problems or whatever. It had produced the same kind of cognitive impairment in terms of memory and attention. And psilocybin is used clinically, so that was quite reassuring. In terms of mood, whereas both drugs made you feel really good, 2CB gave you less propensity to feel bad. So in the sense of people felt less confused, less subjectively impaired, and didn't go down a dark way as easily. And overall, when we looked at the final kind of quality of the experience, we saw that people were describing 2CB as kind of the Diet Coke of psychedelics, in the sense that they felt much more in control, it was less deep, it was very perceptual and visual, and they felt less impaired. So if we go back to this original triangle that I was showing you, whereas psilocybin gives you the full-blown psychedelic experience, 2CB was kind of more in the visual perceptual elements and these like positive mood effects, meaning it was quite a well-tolerated drug. So afterwards, we wanted to see, well, given we were looking at the brain under these drugs, maybe there is something about how these drugs work that can scale up in the brain. They have very different receptor profiles. And so here we tried to see, well, what receptors help best explain these changes in the brain's spatiotemporal organization, so space and time. And what we saw was that, first of all, whereas 2CB produced that state of integration, that whole brain integration, different receptors actually contributed for different purposes. So things like the 2A receptor and the 1A receptor, they actually were more important for like temporal elements of the brain's organization. But then across drugs, when we compared the two drugs, we saw that new receptors, secondary receptors, were really important for explaining differences. With the serotonin reuptake inhibitor, serotonin reuptake transporter, SERT, and the dopamine transporter, DAT, were more really important for explaining the differences between the two. So yeah, maybe pharmacology does scale up in the brain. But given that these drugs affect the brain in terms of space and time in very different ways, maybe there's a way that how they kind of collectively change the brain in terms of these measures or separately that could provide us, let's say, a marker or signature of how these drugs relate to someone's experience. And so that's what we did, trying to see how these measures converged or diverged in a way to explain like someone's experiential profile. And what we saw was that, yes, basically, at the individual level, how the brain reorganized itself in space and time for each of these drugs could help explain non-overlapping elements of these experiences at the individual level. But in this study, right, we were just looking at the person. We weren't seeing how someone was changing relative to his group, right? Maybe there's something about individual differences here. So that's what we did in the next chapter. I think it's chapter 5. Here, we were basically trying to generate a neural fingerprint. So basically, how unique is someone relative to a group of people? And here we conceptualized it as I self, so how unique I am relative to myself in time, and I others, so how different are we all from one another? And what we saw is when people took ayahuasca collectively, their fingerprints blurred, they became similar to one another. And so we wanted to ask, well, maybe there's something in the brain that reorganizes that's unique to each of these people that can help us explain the variability in their experiences. So by ranking the contribution of connections that were most important to define someone and seeing how they would relate to someone's experience, we tried to identify connections that were unique to you that would change under ayahuasca. And we saw that, yes, elements of the brain that explain an individual were really important for explaining the variabilities in experiences. But we have to say that these group of people who took ayahuasca in this study, they belong to a church called Church of Santa Daime, which take ayahuasca a lot. And if psychedelics are neuroplastic, then they have a lot of that going on. So maybe context here matters, not just only in how they take the drug, but also just the whole nature of their style. And so in the final chapter, chapter 6, we want to see, well, if these people are unique, what is it about them that's so special? If they show different functional signatures, maybe the roads, the structure of that brain is very different to the general population, the bell curve. So to do that, we wanted to ask, well, if the structural connections of your brain are the roads onto which the functional connections drive, what we've been talking about so far, maybe by taking a lot of ayahuasca, basically, it changes the roads and therefore changes the nature of functional connectivity. And so to do that, we looked at the interdependency of structural properties of the brain, following the principle that often regions that wire together fire together. And what we saw was that these people, these members of Santa Daime, did have very different brains compared to a matched normal population. They had regions of the brain that were more specialized and others that were more interconnected and diffuse. And when we looked at genes that tried to explain, let's say, neuroplasticity or how ayahuasca worked in the brain, we saw a lot of markers that could help explain these differences. Things relating to cell growth, but also the botanical components of ayahuasca, suggesting that context, whether pharmacological or social, could be playing a role in these long-term effects. So what have I spoken about? So do different psychedelics do different things? I think yes. In terms of across the number of studies, whether it be surveys, lab studies, or fMRI, we see that different psychedelics show different effect profiles, and that pharmacology actually does seem to matter, not only for within a drug, but between drugs. In terms of context, we saw in the Santa Daime people that yes, there seems to be some reorganization, that's common, also in the long term. But remember, these people are really unique. So the only way to know is if we study them in relation to other people who do not really know each other. And lastly, do individual differences matter? I think so, and we seem to show across 2 chapters. But the only way really to scale that is if we scan someone across a range of different conditions. So why did this all even matter? So firstly, there's a lot of interest by clinicians to use psychedelics for a range of psychiatric disorders, like depression, OCD, anxiety, and even disorders of consciousness. But right now, it's kind of like a one-size-fits-all approach. We don't really know what's best for someone. And if I've shown you that different people have unique elements of their brain that helps predict these experiences, but also drugs themselves are very different, then maybe we should move towards a more personalized strategy and start thinking about these next steps for the clinician's toolbox. For neuroscience, like drugs are a really, really good way of perturbing the brain's organization, how it moves through space and time. And as I've shown, different drugs perturb the brain in different ways. So by using these, we can kind of understand the rules by which the brain reorganizes itself to facilitate certain states of being. And lastly, policy. So right now, novel psychedelics fall under this umbrella term called novel hallucinogens. These are substances that have no medicinal use and can put you at risk of abuse, I guess. But I've shown you that 2CB was just as tolerable and just as safe as psilocybin, and 1-PLSD is literally just LSD. So this blanket categorization doesn't really make sense. So instead, we've got to kind of think of the science. So whether it be the chemistry, the structure, or how these drugs work for a more science-informed perspective on things. But ultimately, for me, this work kind of raised more questions than answers, because all these elements, how they interact and what actually matters, is just a big can of worms. And so we have to think about what really is important for the individual. Thank you.
Thanks a lot. It was a very clear summary of your work. And the opposition will now be opened by Professor Arjan Blokland. Professor Blokland is expert in comparative neuroscience at the Faculty of Psychology and Neuroscience here in Maastricht. Arjan.
Thank you very much. First of all, I would like to congratulate you on this wonderful thesis, and of course also your supervisory team, helping you to complete this work and describing all these studies that you did. So that's an amazing job within this period of time. And also the quality and all the things that you described, I think almost like 80, 90 pages of supplements. You just want to show everything, and that's really good for open science, so you should be commended for that. But I mean, the showing all the data could also, we could have all kind of questions. But I would like to start with a more philosophical question, because I think when you're reading your thesis, you also like to philosophize at some point. So when I look at these data and these effects, would you explain all the data, all the effects that you see of these drugs? Are they, can you base them on the biological level, everything, or do you think there is more than that?
Highly esteemed opponent, thank you for the kind words, and no, I don't think so. I think that's the big question, right? Why we have this idea of a biopsychosocial model of things. But really, I think over time, as I've gone on, I've realized it's probably the sum of the parts that's most important. And just finding ways to kind of disentangle things one by one. More in the sense of, let's say you make a model and you have a number of predictors, right? I think even though you won't know exactly what is, there is no singular driver, but if you can understand, let's say, how much something contributes would be quite useful.
Yeah, but so when you only see these effects when you start to interfere with brain function.
Yeah.
So that's a biological, and you see all kind of neurons on your sheets and all kind of interactions between neurons and plasticity, it's all neuronal changes. I mean, for me, that would be enough to explain that.
Sure.
All the phenomenal effects that you find.
But then under these drugs, we have very strong, let's say, there's always this conversation about setting or context or expectancy. It's all synergistic, right? It's all interacting. I can't infer any directionality of what's pushing one another. Let's say these drugs start everything, sure, so it is, but it's just about the magnitude of the outcome. I think that's the question, right? In the sense of, okay, the effect, no, okay. So you're gonna see these changes, right? Because of the drug, there's no dispute. But what you're going to see is going to be very dependent on those other elements, right?
And the other elements are then?
So it could be context, it could be intention by subject.
But context also interferes with your perception. Your perception affects neurons, so it's all biological.
Yeah, sure. Sure. But it's all interacting. I don't know. It's bidirectional.
Interacting with what?
Psychosocial elements, I think. It's not really like a one-way street. That's what I'm trying to say.
Yeah. Okay.
Does that make sense?
Maybe we have another discussion. I mean, so the whole body-mind problem is still not solved by Descartes and still not
the question. No, but I understand. But none of this is possible without the biological effects. Sure, you can be reductionist and say that, yeah, everything stems from biology, right? That's what we are. The drugs act on some pharmacological principle and change the brain function. But in that process, you're still receiving other things, right? The result is contingent on all these other elements. Yeah.
Of course, you can argue whether everything is biological or maybe something
more philosophical, right?
More than that. Yeah. Okay. But that was for a philosophical question. It was no good or bad. Huh? So
yeah.
It's communicating.
Sure.
Okay. But let's go to a little bit more hardwired science. I would like to take you to chapter 3, where you compare 2 doses of psilocybin and 2CB. And the doses are almost equivalent, like 15 and 20 milligrams.
Yeah.
But when you look at the receptor affinity, you see that 2CB is about 15 to 20 times more potent.
Yeah.
But then when you look at the effects, they're almost equivalent.
Yeah.
I mean, you showed in your slides that there are differences, but when I look at the figures for all, maybe you see a little bit higher effects with psilocybin, but I mean, in general, I don't see a difference between the two. But from a PK/PD perspective, how can you explain this much more effective 2CB with the same doses, having this similar kind of effects? What can you explain this?
So already, I think you're touching on something bigger, to me, in the sense of how do you compare apples and oranges in this field, right? So you're mentioning that the 2A receptor is the definition of everything. Here we were matching doses on the basis of just some marker of subjective high, right? So just equivalence of effect. If you actually look at the supplementary materials, I know there was like a lot of them, but you'll see that they're not all equivalent. So 2CB in some dimensions on the POMs, I believe, had greater scores than psilocybin. But this whole idea of equivalence is really hard to touch on because I think to me, after 5 years, I realized that the instruments we use are just really not sensitive. They work really well for distinguishing, let's say, a psychedelic from a non-psychedelic, like an amphetamine. But then within a class, right, where everything is very similar in terms of phenomenology, maybe some distinctions, you're not going to have that much signal, I think.
Yeah, but then I would like to tap on another thing, is that when you compare these drugs and you say, well, it's not always the same, you also mentioned in your discussion that the heart rate was not affected in your study, where it was affected in another study. What was the other way around? I can't recall. But I mean, that's one of the measures that you could not replicate. And so when you measure like 100 different variables, so I can imagine that sometimes you see differences, but and you should have controls for multiple comparison, and then probably the differences between the 2 drugs and all these different measures would be not so much different. Sure. So I would argue against such an
I would also argue against your point about multiple comparisons, because I'm not correlating anything. They're like independent measures.
No,
right?
But when you measure like 100 things, like 5 of them will always be different.
Sure.
Just by chance.
With the heart rate thing, I think it's more to do with the fact that the previous studies are all observational studies. So these people didn't actually have any, it was basically the studies, they were done in Spain. It was people they would invite scientists to their home and literally watch them do
okay, but I think drifting away, my question was about the differences between the potency of the drugs and the same doses that you gave.
So yeah, potency cannot be
explanation.
defined by 2A alone, basically. That's what I'm trying to say. I don't think 2A agonism is sufficient to define potency.
But there's a huge difference, like 20 times potency.
Yeah, but then if all the others matter, as we've shown in the other chapter, then what does that say about matching doses on the basis of a single receptor? But also, you can acknowledge the only way to know is to do a dose ascending study. So the only thing you would need to be able to do is, you would have to do PET mapping, which is still not be done for 2CB. There is no idea of occupancy at all, which is the case. You have it for psilocybin, but not for 2CB. And B, you would have to do a full dose ascending study, which is being done, I think, by Basel. But the question with those is then, it was kind of a broader question of how did you fit everything else in a dose ascending study? You're not going to do fMRI with many, many dose ascending studies, or you can, but it's really costly, and carryover effects are going to be humongous, right?
But I think you explained it well to me, and I will give the word back to the director because of time.
Sure.
Thank you very much, Professor Blokland. I pass on the word now to Professor Franz Vollenweider, expert in psychiatry at the University Hospital at Zurich. Professor Vollenweider.
Thank you, first of all, for this incredible work and presentation. I was really impressed. So, you are already in the middle of the discussion. I'd like me to make it. Just once. That's nice. We are already in the discussion of the dynamics of psychedelic states, and you have shown everybody speaks about psychedelic states, but in fact, when we treat normal subjects or patients with psychedelics, there's ongoing dynamics over time, and so it becomes very complex. What is the right time point to measure biological markers or network alterations, psychological alterations? But my question now is, when we have treated normal subjects 3 times with the same dose, there was large variability within one subject. So it's not between, it's within. So what would you argue, what factors may contribute to these variations? What is known, what has been shown so far, or have you new ideas about that?
Highly esteemed opponent, thank you so much for coming all the way to the Netherlands. I'm really happy you're here. This is the thing about inter-individual variability, because it can happen within a person, right? Even with the brain function, like most of the time, the retest reliability of your brain is very low across sessions. And with psychedelics, I think it's what I was also arguing with Dr. Blokland, is the fact that the contact, everything else that feeds in, is also just probably playing into this stuff. Regarding the dynamics of what could be important elements that play into this, it really could be, let's say if it's a repeated design, right? It could be, for example, if we're talking psychological senses, it could be, for example, people with the integration, the insights they have afterwards, maybe that changes. It could be tolerance, depending on how quickly it happens. But for me, it's really hard to speculate on what's important for a patient, given that it exists within, let's say, a psychotherapeutic structure. So given they have support before and after each of these sessions, I can imagine that the outcomes will change if we're looking at just subjective inventories. For the brain, I think this is one of those questions where we don't really do longitudinal studies yet. We don't scan people, before, during, after, multiple times, right? I would expect that the relative differences that we often see are going to be diminished massively if we have all this additional information. But, sorry, I think I'm waffling.
That's okay. But you pointed in your presentation to the fact that we should use specific neurocognitive measures like emotional regulation or cognitive top-down regulation on the limbic system. These are the things that are studied in depressed patients because they show
I agree with you fundamentally.
Increased self-referential processes.
Yes.
You think psychedelics can interfere with this?
I think also one of my great regrets is everything I've shown so far is all resting states. There's no empirical truth to what's actually happening, what's actually thinking. There's no sort of anchoring of like what it is that people are thinking or what value. I would advocate for, I wouldn't say advocate going back to the days of like bold response tasks. I would advocate for just looking at tasks that are relevant to patients, that are well established, like emotional processing, theory of mind, but through the lens of connectivity. Because I think what I've noticed from imaging now and the past is that really nothing ever sticks when you look at functional connectivity in the brain. But if you anchor it within some form of, let's say, fixed a priori, which is is important for patients, then you can play with that. I think that's why things like neurofeedback exist and so on. But I would agree, yeah, emotional regulation. For me personally, I've always been interested in cognitive control because it's basically the basis of like most important things and also syncs in really nicely with what supposedly psychedelics do, right? But yeah, I think that is the next big question.
Right. One last question would be, we thought that the 1A receptor is quite important because it has opposite effect on the 2A mechanism.
Yes.
And this 2CB apparently is less stimulating the 1A.
Yes.
Compared to 2A, I think it's 1 to 10.
Yeah.
And nevertheless, but it also inhibits the serotonin transporter. Is that correct?
Yeah.
It's quite similar to MDMA.
Yeah.
So, but it's not so much emotional. Is it because of the dose, or have you played around with the dose?
So I haven't, the only data I've seen on dose ascending, often people report like a steep dosing curve, but from the few studies that have been, also in rodents, they often use that step. You don't really see that so much. There's been work by Thomas Polaniczek that says that like 2CB increases striatal dopamine release, which is really cool. But in terms of the idea of tying, like high-level constructs like emotionality to, yeah, it seems to me like, the talk of entactogen and psychedelic, right? I think 2CB kind of just sits there, but we don't really know which one it's pushing more towards without dose ascending. I think the only way to know about the emotionality aspect as well would just be, just do what Lichty did, just give citalopram before or something and just see. That would basically be an easy hypothesis test, I think.
Sure.
Yeah.
And you think that 2CB could be a way out of the problem of blinding of psychedelics, or is it too close to psilocybin? So
that's another question that I have also wondered, because the blinding was like, people didn't really spot it so much. It was like 64%, I think, detection rates between when they were asked to guess. But then I think one of the questions is like, okay, if 2CB is a 2A agonist, if it does the whole brain complexity stuff, if it still produces some ego dissolution, then how do you, is it blinding or is it going to just, it's producing something afterwards, right? So is it a really good comparator? So I would call it an active comparator more so than anything. But yeah, so for now we've started looking at the data afterwards, because after each dosing day, people reported questionnaires 1 day and 5 days later, and people do report positive social effects, positive mood effects, better mental well-being, and this like small time span in a way that's equivalent to psilocybin. The only thing that changed, I'm sorry, this is not out here yet, was the meaning. So in the persisting effects questionnaire, people didn't rate it as, the most significant experience I've had in the last month, in the last year. It was not that high.
But blinding in terms of, let's say, how FDA thinks.
Oh, yeah.
It's more, it should mimic the psychedelic experience, but should have quite a different neurobiological basis.
Yes, I would say so. But I think the only way to know is we do the longitudinal, single fixed dose high, do the typical MEQ, all those, because we didn't do the MEQ, but all these typical FDA pipeline kind of questionnaires, and then just see, and at that point it would be interesting. But then, to me, it's hard to say because it hasn't been studied appropriately in this typical like pharma way. But I think the way to know is, the alternative is just sometimes, I've seen it used before in Switzerland in the 80s. They were using it as a sort of adjuvant, right? So people who were scared of losing themselves or ego dissolution or whatever, I think it's also advocated that MDMA could be like an introduction. But the problem with MDMA is it doesn't really produce that loss of self. So if 2CB produces that loss of self-referential awareness without the capacity to potentially feel bad, then that could be a useful practice for those who are neurotic, young, and so on. But I think since this is just one study in a clinical context, with active comparators with mostly just students, people 18 to 30 on average, it's really hard to project long-term from this one work.
Okay, thank you.
Thanks a lot, Professor Vollenweider. I pass on the word now to Dr. Katrin Preller, expert in pharmaco-neuroimaging and cognitive emotional processing at the University of Zurich. Katrin, you have the word.
Thank you so much. Also, I would like to congratulate you on a fantastic thesis. It really was a pleasure to read. My first question centers around the fact that you have now experience with and built a lot of expertise in a number of behavioral as well as neuroimaging techniques, as well as data analysis methods, which is really nicely showcased in your thesis. So thinking not about the past, but more about the future, have you discovered some techniques to be particularly useful or not so useful moving forward? Because you said in the end of your talk that maybe we haven't quite decomposed the psychedelic state yet, right? So what is it that you would move forward? What is it that you would do differently to you know, eventually hopefully get to a decomposed psychedelic state?
Esteemed opponent, thank you also so much for joining in. It's really a pleasure to have you here. Yeah, I thought about this a lot. I got disillusioned as time went on about just resting state fMRI and static functional connectivity. I think so far nothing, that there is work that's ongoing in consortiums, right, that shows that nothing replicates locally. And these questions will only get worse when the variability of our sample is increased, right? As we recruit more patients from many different types, the value of neuroimaging is just going to drop because we're using measures that are really coarse. And so what I've recently it's been going on for a while, and I'm not the first. It's all this idea of like nonlinear dynamical systems, right? In the sense of basically, often what you see, there is this final effect that's really difficult to understand. And there are regional interactions, but they basically scale up, right? So this idea of using complexity or these univariate sort of linear measures, right, which is like one-to-one. First of all, no region in the brain is operating one-to-one, right? It's simultaneously connected to others. Secondly, defining regions, you know, based on an atlas is also very difficult because people have very different brains. So what I would kind of advocate is more looking at basically these methods that are taken from the field of statistical physics, which is just kind of understanding the general change of something rather than trying to isolate, let's say, individual regions. Also scanning a few people a lot around many different scenarios, not using resting state. I have so many, but it's like, I think most of the time, you know, to try and like replicate resting state is difficult because everyone says different things or thinks different things. So when you average all of that out, you know, you don't find anything at a local level. So what I would just suggest is just looking at the totality of the effect, finding, so I see a lot of work with like, you know, attractors, you know, defining these sort of supposed neural landscapes, but you could do it in a closed loop system. So basically you show someone, you start scanning and you expose someone to a range of different conditions that you know are clinically relevant or behaviorally relevant. And how someone moves through that space is a really good indication of, first of all, what is it that they're actually thinking about doing, and it's constrained. So in the question of like individual variability, okay, people are going to move slightly differently through that space, but they're not going to go off in 20 billion directions like rest. So you have this sort of shared effect. Also, if you scan one person lots of times, you don't have to use some parcellation that's kind of randomly defined. You can just make your own parcellation from that person, right? So there was a paper, the Nature paper, I think, which was just kind of a beautiful representation of that. The precision scanning this by Siegel. Yeah, that was really amazing. Sorry.
Thank you so much. I would love to have the time to discuss that for another 2 hours, but I really want to ask you my second question as well. So in your thesis, as well as at the end of your presentation, you mentioned that all of this work is important for clinicians to take into consideration. And I would like to ask you, like, can you elaborate on this a little bit more? Like, what can we learn from the work that you have presented for clinical practice, or what needs to happen so we can learn from it for clinical practice? And how are you envisioning these personalized treatment approaches that you're mentioning?
Thank you for the question. I think, unfortunately, I don't think this work alone can change clinical practice, but it can change opinions on it. I think for me, there is this whole idea, right, of one size fits all. But the one size fits all approach changes in terms of the practices around the drug. But for now, I think we pick drugs based on clinical ease or tolerability, right, in the sense of maybe we'll give LSD in certain scenarios, but now people want to give DMT because it's quick and it will take care of like a lot of the issues. But if we have a better understanding of, first of all, at the patient level, right, what defines a symptom profile, like a fingerprint of a symptom profile, or what defines, you know, a drug's effect profile, then I think we can basically foresee a world in which, you know, you have some understanding of baseline qualities of someone that you know would be better suited for a certain drug. In terms of actual immediate applicability, I just like the idea of 2CB being added in the conversation, in the sense of, I think there is always this conversation of kind of psychedelic exceptionalism, right? Especially you see it around certain drugs. But if you see that there is a broad range of different psychedelics that seem to do, let's say if the brain is actually important, and I think, yeah, if there is a broad range of different psychedelics that kind of do the same thing, then why not kind of start exploring different pros and cons, the relative advantages and disadvantages of different psychedelics. So in terms of clinical use, I think it's just a step in that direction. And also this kind of first idea of first comparing these psychedelics, right? Typically we just compare, you know, let's say MDMA and LSD, but we have to start asking ourselves now in a set group of people, what are the relative pros and cons of different psychedelics, I think. And neuroimaging can kind of help us, right, in that sense, I think.
Yeah, no, thank you completely. I completely agree. But still, kind of like, well, if we want to personalize our treatment, right? We kind of we need to link different things, right? We need to link the specific person, to either a drug or a dose or context, etc. So, how do you think that, you know, for example, the neuroimaging work really fits in there, and what can we learn to actually get the best treatment to the patient?
I think it would depend on patient characteristics themselves, right? In the sense of, let's say, I'm sorry if I repeat myself, but let's say some patients have, let's say, a specific, let's say, comorbidities or so on. Let's say their symptom profile is mostly related to rumination or something like this. Let's say in an ideal world, we have a good understanding of rumination. And let's say we scan them and we're able to get an idea of, let's say, the underlying, what's it called? That underlying kind of marker of that. So what I'm just thinking of is the Menage-Dos paper, in the sense of they show that baseline neural flexibility is really important for predicting, let's say, improvements in cognitive flexibility. So if you have patients that say you make them do a battery of tasks that you know is well validated and has some kind of neural kind of underlying thing, then maybe this could be a way of being like, oh, you're probably better off with this, or let's say you show low neural flexibility, so maybe psilocybin could be for you, and so on. And then at that point, you could distinguish between drugs in the sense of, well, if this person, let's say, has a generalized anxiety disorder as well, or then you could probably think about the kind of different treatment options that are there. For example, drugs like DMT may be too overwhelming for people. I think it's not just the neuroimaging, right? It's like a clinician sits down and thinks about the person, the life, hopefully, the neurobiology, and kind of makes a choice in that toolbox of those that kind of decides.
It does. Thank you so much. I can't see the other people in the room right now, but tracking time, I think I may have used my 9 minutes.
You would have time for a short last question, if you like.
That's fine, thank you.
Then thanks a lot. I pass on the word now to Professor Rainer Göbel, expert in cognitive neuroscience, Maastricht University. Rainer.
Thank you very much, Pro-Rector. And I want to first start by also complimenting you for this amazing work you have done, and also your supervisory team. It was really a pleasure to read your thesis. I learned so much. I'm not an expert on psychedelics, but highly interested. So in that sense, for me, it was very exciting to see your introduction chapter, your discussion chapter, but also all the pieces of good work you have done. Very impressive. And actually, some points were especially relevant also for my questions, especially on resting state and what else you could do. And I come back to this. But before that, I want to ask you about some more small details, because what I was also impressed about your work is the different levels you master, right? You go from the tiniest methodological points up to the global questions about psychedelics and so on. And this is impressive. You don't see this so often. So I want to check a bit how much details you have, knowledge you have. For example, on your resting state chapter, you follow quite standard, mostly pre-processing advices, but still, I was not sure whether you thought about why you use some of those. For example, I could ask you about all these pre-processing steps, but I want to come to more challenging questions. But let me first focus on just one, namely the high-pass filter you used. Oh. You used 0.09 to 0.08 high-pass filter, which is actually, at least the low-pass part, quite common in resting state. But people discuss it a lot over the years. And I want to ask you, can you motivate why you use that filter?
Highly esteemed opponent, unfortunately for you, I have a very boring answer that is not, it's basically just motivated on prior work and replicability. But you're right, I have to say there's more and more questions now. For example, now when we started looking at sort of more dynamical analyses, like what does it actually mean? Is there information there? Sometimes people only use high pass. This is, I think, is the big difference between like methods neuroscience and let's say clinical pharmacoimaging neuroscience. We tend to use bog standard templates without really thinking about it.
But this is important for some of the results, of course, because if you have these filters, you basically filter out everything which is longer in duration, has cycles below 125 seconds, which is okay because most people in resting state say we look at infra-low oscillations in a certain range, right? But for some of the work, it could be interesting to look also at more slow oscillations. The other point is your high-pass filter filters everything out which is faster than about 10 seconds, actually 11 seconds. If you use just these numbers of hertz, right? And that means things where you might have coupling in a bit more task-related or more interesting way for some other questions, you completely filter out. So therefore, I just want to say, you can argue for that filter. It's a good one. Not saying that, but it's good to know why you use these. Right? This is the point I want to go to.
I agree. No, you know, so in terms of the low pass filtering, it's like we were basically just trying to. The idea is just physiological noise, right, or like scanner drift and whatever. But no, I fundamentally like in the future, and I see people moving. It's just we would like to have basically more. We do collect it, but we don't really use it fully. It's like respiratory belt, all these other things, pulse oximetry, pupillometry, all these things. But you're right. No, you're fundamentally right. Sometimes the way that I pre-process some of these studies is kind of in the generalizable way, in the sense of I can repeat the analysis and see. So right now, the work that we're doing is part of a bigger consortium where we just play with all these pre-processes. So whether it be global signal regression, what is the filtering, frame-wise displacement, is it anatomical ComCore, or is it ICA?
Exactly.
These are all really important questions, and they show very, very different things, as soon we will show. But this is,
But this is okay, because that goes directly in the way to make you aware of this, that even if there are standards, they are not set in stone, and you can change them if you have specific questions where you should maybe look at their value. And you seem to have done this in this work you just described.
I agree.
I want to move on to a few more questions which were alluded to already, because I also wanted to not criticize, but want to say, are there not other opportunities than resting state fMRI, right? And also the trend these days, you also alluded to that, like deep scanning, having individuals more longitudinally following them over time and over many repetitions of certain studies. And my question to you, first question is, if one would use task-based fMRI, which I want to use in the future with drugs, talk to you about this hopefully tomorrow. But my point is, what would be an interesting task-based study you would want to do and why?
I have so many. For me, really, so in terms of task-based study, I think I mentioned 2 examples already, but they all basically just probe into the same construct. So I'm really interested. I think there's this whole idea that your brain is more flexible. I don't believe that. It also doesn't really, a little bit, but it's not non-specific like that, right? You don't just get rid of everything. And often what I find is to do tasks with people who are very high is that you have to treat them like children a little bit. So you can't make them do like a gambit task or something like this. So really kind of the future of things for me are more kind of ecological paradigms that span lots of different conditions. So it's not really about the outcome of a specific task, it's about someone's movement through tasks, right? If that makes sense, the general kind of impression that they leave behind. So for example, if you look at the field of like developmental neuroscience, right? Films are really good. Films are really good, first of all, because what I was saying about having a ground truth, you have a ground truth for the scenes, but also people move less, right? They're focused on it, and it constrains this variability. But then in the context of clinical imaging, then that's kind of custom. So I think I mentioned this in chapter 7, but like perpetual multitask designs is kind of really flexible. So let's say you have a really, my interest has always been kind of like social cognition. I haven't had time to put it in this thesis, but there are stuff I'm doing on it. But it's kind of, well, let's say devising a battery of tasks that you know are orthogonal, right? These states are very, very orthogonal. But that are clinically relevant. So you could look at things like, you're moving from, let's say, some social scenes, or like people working, like social exclusion, things like this, different kind of conditions that fit a general kind of RDoC criteria, and understand what is, rather than the behavioral outcome on a task, or their actual performance on a task, how they're shifting from tasks. So in terms of the films, it's very obvious. It's, for example, if we watch a film, we could go from puppies to like a funeral scene. That transition, right, which could happen multiple times, is probably more interesting and relevant as to how someone moves through life, I think.
Yeah.
So
very good points. I want to, actually, this is nice that you have these many ideas indeed, and also they show it depends a lot on the questions people have when task-based fMRI is good and important. I want to combine task-based now with one other point where I felt the frustration of yours, which I felt 10, 20 years ago, namely the limits of neuroimaging, right? And I think one way we try to push this further is what you used also, high-field imaging. You used 7 Tesla a lot in your work. But you didn't use it in the way I would want to use it in the studies I have in mind. And this is very important for, in my opinion, for better understanding psychedelics, so it should be also important for you, namely to go to a laminar level.
Layer 5.
Because if you look at your visual loadings there in your results, how strong the changes are there, and you look in psychiatry, computational psychiatry, where, for example, predictive coding has specific theories on top-down and bottom-up processing, gain control, precision, how this might be changed by drugs could be studied in a way, I think, where imaging would shine. What do you think?
I fundamentally agree. I mean, I was always interested in layer fMRI. I think Renzo, because we have such testable hypotheses, right, in terms of we know that layer 5 pyramidal neurons are really important, but we also know that destabilization of these local populations can lead to broad changes in like whole brain dynamics. So that's just already like a really easy, like neurobiologically grounded perspective. Is like how much does layer 5 actually consequently like affect the rest of network dynamics? But I've always felt, they say that when you do a PhD, it's like your teammate is you 4 years ago, but you were really bad. And it's true that I have often had the feeling that we've used the 70 almost like a free T, in the sense of we have the advantage of the signal to noise, but we don't make the most of the full potential spatial resolution.
But both is important. I mean, we also do questions on neurofeedback, which sometimes use a 3 Tesla scanner at 2 millimeter.
Yeah, no, both are useful.
So it depends on the questions. But if you have questions to understand psychedelics at the laminar level, and I personally have those,
I agree.
then you need the other thing where you have more noisy data, but therefore you have more spatial specificity to separate, for example, the different, at least 3 compartments of laminar effects in the brain, which might be crucially interesting.
I have a personal dream of just kind of building an integrated connectome, so structural connectivity, functional connectivity at rest, task-based fluctuating connectivity, layer MRI, and chemical shift imaging as well, to basically have this idea of like layers of organization that stack. And with layer MRI, I think it is the most essential question for these drugs, because there is a strong circuit-level inference, also for areas like the claustrum, high.
We just studied this in a collaboration with a neuroanatomist. Claustrum is super important. But I think my time is up. But I want that you continue the stream. Thank you.
Thank you.
Thank you, Rainer. And it sounds actually that you already have your next 50 years of working plan ready. Last but not least, I pass on the word to Dr. Peter Stiers, expert in neuroimaging at the Faculty of Psychology and Neuroscience. Peter.
Thank you. Yeah, thank you for this splendid work. Of course, also the team, because I know it's not easy to do this kind of studies. I try to imagine what it must be like to put a person under the influence of a psychedelic in a scanner. I did adolescent studies, and that was already very daring. So this must be really a complex, organizational complex thing. Also, I was really impressed by the state-of-the-art acquisition and, of course, analysis tools. I was so pleased that I would almost say that it was agreeable or pleased to read it, like Rainer said, but actually it was rather hard to read. So there's a lot of technicality, and which is a good thing, so I'm not criticizing that. But yeah, so thank you for this really high standard quality of work. I have a few questions. Some of them have been already addressed, so I'm not going to make a kind of selection. There was one thing that I noticed, actually, when I thought about psychedelics in the scanner, I thought, yeah, these people are going to move so much, and movement is really a terrible source of noise, so how is this, what's going to come out of that? But then I found out that, particularly on the 2CB measurement, that they tended to move less than in the placebo, which is kind of surprising. I don't know what your experience with that is, but I also noticed at a certain point you give a density map of the 2HT2A.
5HT2.
Sorry,
Okay.
should have read it. Receptor, and then I see that particularly the sensory motor cortex has less expression of this receptor. Do you think that there's any relationship to that, or maybe in general, could you predict from a particular distribution of neurotransmitters what who's going to move more or less?
Highly esteemed person. Highly esteemed. Thank you for that question. So with the movement, people do tend to move, and we quantify it in different ways. So it can be frame-wise displacement. The doses that we use are always moderate doses. So people are already psychedelic experienced, so they kind of know what's coming. And also we train them beforehand, so they're familiar with the scanner. I think those three elements, whether they get trained, they have prior experience, and its moderate doses means that actually people don't move so much. It is the big question of pharmacoimaging, how to stop people moving. Regarding PET and fMRI, I have to state that the PET data that's collected are transcriptional template maps. So these are collected by some external center, and I tend to use these as weights, basically, for any analysis. So it's like a ground truth a priori. I'm not sure if you saw, there should be a lot of 2A in the sensory motor, but it could be because of the image I used, I wasn't scaling it. So normally I would z-score it, and then you would really see the sensory motor cortex has a lot of 2A. Regarding predicting movement with that, I'm not sure. Given that it's just a general template, I'm not sure it would match so much with the individual subjects. Seeing who moves or not is a hard one. Really, we can't predict it. It's also why you also often get so many dropouts. When we use the moderate doses, we don't really lose that many participants. I think across studies, the imaging studies, there's only like 2-3 people on average that drop out. But when you start using doses of like 25 mg psilocybin, 30 mg psilocybin, I have a friend who's like doing DMT stuff. Like the dropout is humongous because of this movement. And that's a question that no one really knows how to answer, because the higher the dose, the more exciting the brain is, right? But we don't really know. I think it's just vetting people, but if you vet people, then you're self-selecting your samples. I don't know. It's the one big unknown question.
Maybe you can consider the bite bar. When I started imaging, we scanned everyone with the bite bar, so there was in the coil something sticking out, and you had to bite it, and then.
Oh, that's good. I didn't even know that. I've seen dielectric pads. Sometimes people use them as well, just to improve like signal to noise. But when I've had them on my head, they also do kind of help to not move.
Well, I can assure you, a bite bar is not pleasant, but it does work well.
I wouldn't want to share that one with someone.
Maybe another question, which is not unrelated. That is, let me see. Maybe discussion. What I'm a bit surprised is that's what most people do is they have a placebo condition, so they have a normal brain, and then they compare the same brain or under influence of a substance. But would it not make more sense to see the brain when it's under the influence as a state of its own, and not superimpose on it or ask questions about it from the perspective of the normal brain? For instance, the brain has been divided into several subnetworks, and everyone uses these subnetworks to group voxels or smaller areas into larger areas. But would it not make sense, for instance, under a particular drug to do a completely new parcellation and see what would be the best description of the structure of the brain under these under this influence? Or, for instance, use completely random models as a baseline instead of the real brain, or maybe anesthesia data or something like that?
Yeah, that's a really great question. So actually, independent components analysis, right, on the actual like state. But then the problem is if network structure gets like completely disintegrated, I've noticed that it just basically produces like very small little parts.
You can shortly finish the answer, because I'm also curious.
So if you do an independent components analysis on someone that's on a psychedelic, or even sometimes if you do it across multiple conditions, it doesn't actually ever, you'll just end up with like DMN, A, B, C, D, E, F, G, H, I, J, K, basically just saying everything is disintegrated. So it doesn't have much use in that sense, I think. But if you scan someone over multiple time points within the same day, maybe you could have some form of latent signature, but then maybe it's just probably better to just do it across conditions.
Okay. Thank you, Dr. Stiers. Pablo-Alexandre Mallaroni, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I kindly ask you and the audience to await our return. We will come back for sure. Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. I know your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Where's the TA result? That seems like a really important thing. That is the big one. That's the one. And also, I thought the way this couple really likes it. Yeah, but it's people could have handed it to So the way he prompted you at the end, I thought it wrapped it up really nicely. Where you were talking when you spoke speculatively about the way that you want to act, the imagery. Yeah, I just thought it was all wrapped up nicely. Yeah, very. Yeah, no, yeah. We're the same. I didn't realise that you face limitations even with like the sharpest tools. Which is where the filters come in, right? That's all. It's the same thing. You can see dog shit on the pan. It's alive. It's alive, right? I like this. Very like you got string on there. Pull it out. I only got this one. Oh, that counts as the best. Wait, I don't. So few no pressure. see what happens. Anyways, I've got a bed.
Oh, the monk as well.
Stay okay. It looks. I haven't seen the one with the one with on the river. Oh yeah, next to the bridge. Yeah, the big red there. It's right on the river. It's really nice. Like the building is well located. Yes, over the side of the bridge. Yeah, no, it's fine. I mean, it looks really clean. I don't know what to do the same way. Check it out. Just got it. Yeah. I thought that was good. I didn't even yeah, this guy's smart. Holds the room. It was a hard, I remember it being a hard question. It's like those two, the machine isn't sitting at the line to tell you everything. Just got to. It's good.
Pablo-Alexandre Mallaroni, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Ramakers is authorized to confer upon you this academic distinction in accordance with the Dutch university custom, and I kindly invite you now, Jan, to take the floor.
There we go, Doctor. And do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Pablo-Alexandre Mallaroni, cum laude, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Pablo, this is the time for the laudatio and to think back a little bit. And I was thinking, how can I describe you in one word? Now I would think maybe two words would be better, cum laude. But the word that I actually came up with, the first thing that came to mind was complexity. And for many reasons. And I would like to go through the list of complexities that I've encountered with you over these last 4 years. And the first one, of course, is obvious. It's the complexity of the thesis. People have mentioned this already. There's a broad scala of topics and methods and approaches that you've shown to master in your thesis. But the list of measures that you worked with is really impressive. And we work with classifiers, and you introduce psychotropic dose equivalents, and you have numerous measures of brain complexity that you've been working through all these chapters. In addition to that, you didn't have very easy study populations. Healthy volunteers were maybe easy, but on drugs it's always very difficult. You had the Synthesizing, the entire Dutch Synthesizing community in a scanner at Maastricht University, and you scanned 24 people in only 4 weekends, on ayahuasca and off ayahuasca. I think that's really an achievement on its own. And I also remember when we were discussing the design of the 2CB study, and Natasha was involved in this as well, I think that the design itself, thinking about the design, took at least 6 months, because you always came up with new ideas. Yeah. And you alluded to it already, but in that study, we also had task-based fMRI, right? We had tasks on social cognition. We had tasks on emotion attribution or re-attribution that you're still working on. We had the sub-acute data, where we looked at the impact on aggression. We have cytokines data. So I think there's still enough data out there that you potentially could fill with a second booklet or like a second thesis easily. But the fact that you pre-sort all of this at a very early stage actually means that we can now profit from all of this thinking collectively very well. And then also, and the final thing about the thesis and the work, because most of it has been published, but that publication process or going through the reviews has been very complex in many cases, and particularly when it was about the neuroimaging work that you did, the functional connectivity, because then you actually get confronted with a lot of insecurity in the field. Because if you make a choice for a certain brain atlas or a certain parcellation or a decision on whether to include the global signal or not, then there's always somebody who disagrees. I say, well, maybe if you use a different parcellation or a different brain atlas, it would look totally different. And then I would actually give you that as an assignment, which are very difficult to do because you can't do it overnight. It should mean that you have to initiate your laptop and wait for 3 months for the data to come out and then go back to the review process. But you manage your way through that, which is very admirable. So then the second point I would like to stipulate is like your complexity of thinking. And this is where we get to the wormholes, Pablo's wormholes. And wormholes are known from Albert Einstein, but when he spoke about shortcuts in space-time, basically meaning that you can travel, you have shortcuts to go from regions to regions. But in Pablo's wormholes, there are no shortcuts, there are only dead ends. And that's a little bit a problem also to yourself, because you hate to be in wormholes, but you're actually always in a wormhole, it seems. And I remember at least one wormhole when you had collected all of the data of the 2CB, and you were very excited to do a comparative analysis on your functional connectivity data. And all that your computer showed was there was no difference whatsoever. None. There was no change. And of course, that could not be true, but this is a wormhole or a problem that survived for at least 6 months, I believe, before you actually found out that it was a very simple parameter setting somewhere, in the small prints that you overlooked. And once you had set that right, all of the data started shedding in. So imagine, Pablo, we could have been standing here 6 months ago already.
Probably.
Probably. And then this complexity of thinking also transfers into the way you write. Because the first time I saw your manuscript, or sometimes I thought, what am I reading here? Is this a scientific paper or is it literature? You're the only PhD that I came across that actually made me look up words in the dictionary because I couldn't understand some of the adjectives that you were using all the time. And you seem to think that the more adjectives, the better, or the more precise actually the descriptions are. So, but in this complexity of thinking, you're never really alone because you know that you need other people to sort of come out of the problem. I mean, you do always solve the problems, I have to say that. But you have collected like a nice network of nerdy friends, I would say, that you can call and contact and discuss all of your scientific issues with. I'm thinking, for example, maybe some of them are online, I don't know, like Manoy Dosh. You mentioned the name of Greg quite a lot, and Jeremy. Mr. Flip-Flop has been in the negotiations quite a bit. And throughout your time here in Maastricht, we also set up collaborations with our friends in the Coma and Cognition Group in Liège. We have this collaboration with Paris. So your network of expertise actually has been expanding quite a lot. And you're using that actually very well. And people like to see you actually in these networks because they understand that you're one of them. You're one of the nerdies. and which is a good thing, I guess. And then the complexity of personality, Pablo. That's the third one on my list, and that actually starts already with your name, right? Pablo Mallaroni. So when I first met you, I thought, he's from England, right? Everybody who hears Pablo speak thinks, he's from London. Which makes sense because you have been living there for quite a bit. But then you think, okay, but then how do you get to a name of Pablo Mallaroni? And I thought, well, maybe there are Italian roots, but no, it's Corsica. You're a Frenchman. And people would not actually think that very easily with you. And then there's the name of Pablo, and then there's a connection to your mother, who is from Mexico. So, and hence, you speak also 3 languages. You speak fluent English, you speak fluent French, and I think also fluent enough Spanish. I look at math, you see, your mother agrees. So that's, that's a complexity of roots in a way, right? And then also sometimes I think you will wonder yourself, what am I? Am I French? Am I Mexican? Am I UK? Or just a Maastrichtenaar? That's also a possibility, of course. I don't know. And then there's Pablo in the social context. So I think the family here on the road knows a lot more about that. But I specifically instructed Pablo today not to wear headphones, because that is his favorite position, to walk over the corridor, sit behind the screen, and wear his headphones. And so he likes to be in a social isolation, and he chooses to be there quite often. But at the same time, then, if you invite Pablo over for a drink, then he doesn't stop talking. He just doesn't stop talking. So there's this weird controversy between him looking for silence and no communication, and the other extreme opposite of just constantly talking. There's nothing in between. So that's that adds to your complexity. The other one is your choice of dress and what you wear, Pablo. So the motto is black is back, I think, is your motto. Or never was away, never went away in your case. And even see that you instructed here your paranymph to also wear black. This is basically your coat. So well done, Pablo. You got you're spreading a style here. And I remember it was like 2 years ago. We were moving to a conference in the United States in Denver, but we started our trip in Las Vegas. So we rented a car. We drove through the desert, and we also walked in the desert. Everybody was about butt naked because it was so warm, except for Pablo, because he was wearing his black boots and his black shirts and his black pants and his black leather jacket and even a black hat to protect his sensitive skin against the sun. But he would not change his style whatsoever, not at all. And then, and Pablo's also very attached to his laptop, right? After 1 year or so, I think you came complaining with me that this stupid laptop I have is really not powerful enough to do what I want to do. So we bought you the strongest, most powerful Apple laptop we could find. And I think it took you only 2 weeks to actually crash it by just going into a plane and put the laptop and the table in front of you, and then the guy in front of you pushed back his chair and crashed basically the screen of your laptop. That was a big frustration for you at the time. But then, so there's complexity of your personality, but there are sides of you that you do not show to everybody. And I sometimes have the impression that you really feel liberated only when you go to Japan. No, that's partly, of course, I guess, because of your girlfriend. But you tell me many times that you like karaoke, which is true, right? Let's see. And so I remember Pablo and I went to a vinyl shop maybe like 3 years ago, and to buy some records. We like to play some of these old records. And then Pablo bought a record because that he liked so much and that he wanted to sort of practice for in a karaoke session. Do you still remember which record that was, Pablo? Did you all hear that? Serge Gainsbourg, Je t'aime moi non plus. Are we all old enough to know which song that is? So, I have never ever heard Pablo do a rendition of Serge Gainsbourg, but we're going to hear it tonight at the reception as part of Pablo's determination tonight. So then finally, Pablo, there's, this is my fourth point, the complexity of the moment, I call it, and looking forward. And that's basically, now that you have finished your PhD, what is next? And how did I get here? And I look through the acknowledgement, and you thank me and Natasha and Enrico for their help, for empowering you to become the person that you are. And we thank you for these compliments, but in the end, I think it really was you who empowered yourself. It's not us. And to stay in psychedelic terms, I think the three of us, we provided you with the sets and the setting, but you not took the trip all by yourself, and you made it happen, and you brought this project further than we could have ever imagined 5 years ago. So the empowerment is all on you, Pablo, and you made it happen all because of you. And then, where to go now, right? This is another wormhole, basically. What you wouldn't be Pablo if there would be a new wormhole that makes you think and wonder about your future, your sense of belonging. We spoke about it already a couple of times. And that's really, where do I belong? Right? Should I go to Paris? Should I go to London? Should I stay in Maastricht? Should I stay or should I go is a very famous song in that context, and I think it applies very well to you. And it really comes down to this question, right? I know you want to stay an academic, but the question is, where will I find these opportunities? And I do understand that at the age of, at the famous club of 27, and you're young and you still want to contemplate and make the right choices. But I think, I didn't ask, but I dare to say that every academic, all the professors that are sitting here, right, they never planned their entire career, because that's basically impossible. So it's not possible to upfront think about what are the right decisions. It's all about occasions that come by and that you have to grab. I think the only thing that you probably can do to stay in psychedelic terms again, is to think about your priors, right? What are your priors? And set them in the way you like them. And then make the choices of opportunities that come by. And I'm sure they will come by, and I'm also sure that you yourself can create opportunities in any way you would like them to see, and in any way that they match with your career and also with your private environments. So priors are important, and know you understand what they are, and I think also know what they are. So don't be afraid to stick to them, and also say no to opportunities if they do not meet the priors. So Pablo, I think we are all very, very proud of you. And I thank you for the collaboration. I hope we remain collaborators. I think we will be also in the near future. I thank you. I congratulate you. Also the family, Mia, all of your friends, everybody in the audience with your success. And with that, I give back the word. Thank you very much.
Yeah, thank you, promoter. Also on behalf of the rector and the dean of the Faculty of Psychology and Neuroscience, congratulations, be proud and enjoy this day. I have a formal remark that there is no reception in the rafter, but we have a short congratulation round behind the stair at the main hall. Go there already, that's my suggestion, and shake hands. We will meet, like the people here will meet at the staircase to have another photo moment. And we will have a short photo moment here, first also in this room, because then Katrin is still online, and it's for you a good memory to have Zurich with us. So Katrin, please stay. I kindly ask the audience already to leave and enjoy, and especially enjoy this day and enjoy this great work and this great human being standing here as a doctor cum laude. I formally terminate this academic session.
Oh, sure.
Yes.
Go with you and just come a little bit forward so we can take a picture. You, Magnus, and Commission.
From the bottom,
Please come a little bit forward. So one of the photographers can make a picture with you and the online opponent as well. Supervisors